So in our study, we were very much interested how the different cell types of the brain change while we age. This is very important because we live in an aging society, which means we are getting older and older. But this also comes with an increased incidence of neurodegenerative disorders, such as Alzheimer's disease, for example. So we really wanted to understand what happens as we age. And in order to do so, we obtained brain tissue samples from individuals that had decided to donate their brains to science after their death. And what we then did is we used a technique called single nucleus RNA sequencing, where we first extract the nuclei of the brain tissue, and then using this technique we can examine the transcriptome, so relating to the gene activity of the different cell types in the brain. And what we found was first that all different cell types in the brain change their gene activity throughout the process of aging. This is probably not very surprising, but what we specifically found is that there was one cell type that seemed to be much stronger affected compared to other cell types. And this was a specific subtype of inhibitory neurons, characterized by a specific gene expression profile, which includes the gene LAMP5 and LHX6. Then focusing a bit on like Alzheimer's disease, we were very much interested in, okay, how do our age-related changes that are associated with normal aging relate to changes seen in Alzheimer's disease? And what we did is we compared our data set to another data set from the literature and saw that there's really certain age-related changes that are also seen then in Alzheimer's disease. And what we think that this could mean is that there are certain genes that change their activity throughout aging, but at a certain threshold they become relevant for a disease such as Alzheimer's. And then finally, we wanted to see if potentially the aging process might be different in individuals that suffer from a psychiatric disease. And in fact, we found evidence for accelerated transcriptomic aging in these individuals, which could be interpreted as these individuals are biologically older or they age biologically faster. And what we then additionally did to get a bit better insight is we compared again to another data set that was published in the literature, and we found again that there was an overlap between these age-associated genes that we found and the schizophrenia-associated gene expression changes. And this could potentially explain on a molecular level why, for example, individuals diagnosed with a psychiatric disorder have also a higher risk for later on suffering from an age-related disorder such as Alzheimer's disease.